Hereby, I open this academic ceremony in which Adam J. Sellers will defend the academic thesis, Physiology of Cold-Exposed Humans and the Implications for Type 2 Diabetes. May I invite you to present a summary of your study and the conclusions of your thesis.
Dear Pro-Rector, members of the Corona, family, friends and colleagues, thank you for attending my defence of my PhD. Obesity is a serious problem. More than 1 billion people worldwide are obese. Obesity results in disorders such as type 2 diabetes, cardiovascular disease, and cancer. In particular, type 2 diabetes is characterized by elevated blood glucose levels. After a prolonged duration, the blood glucose can cause damage to nerves and blood vessels. Well-known lifestyle treatments for type 2 diabetes include exercise and a healthy diet. For my research, I investigated the effect of cold exposure as another potential treatment. The research I performed consisted of 2 parts. The first part, I performed 2 experiments investigating if cold exposure can improve blood glucose control. For the second part, I studied the long-term adaptations to cold in a unique population. Before presenting my research, I will introduce some relevant background information. The body aims to maintain a core temperature of around 37 degrees Celsius. This is because our organs work well at this temperature. During cold exposure, blood flow to the skin is reduced. This reduces heat loss to the environment and will help to maintain a core temperature. Also, energy expenditure or heat production can also be increased during cold exposure. When we are resting at a comfortable temperature, the body uses a consistent, relatively small amount of energy. During cold exposure, the body can generate more heat or use more energy. The body can only increase heat production by a small amount without shivering. However, with shivering, heat production can be increased by a much larger amount, as shown in the figure. So, how does this cold exposure relate to type 2 diabetes? Well, previous research before my PhD found that exposing patients with type 2 diabetes to a cold room for 6 hours per day on 10 consecutive days improved blood glucose control by a large amount. This was a remarkable finding and resulted in follow-up research. However, when the experiment was replicated without shivering, during the cold exposure, there was no improvement. So these 2 studies suggest that shivering is important for health. This leads to the first part of my research. Can cold with shivering improve blood glucose control? For my first experiment, 15 healthy men shivered for 1 hour or rested for 1 hour. Then, 90 minutes later, an oral glucose tolerance test was performed, which measures how well the body can process a sugar drink, a glucose drink. Here is an example of the cold exposure. Participants were wrapped in a water-perfused suit, and the temperature of the water was decreased to 10 degrees Celsius, and then participants shivered for 1 hour. So then, after the shivering, participants consumed the sugar drink, and then blood samples were taken repeatedly for the next few hours. Here are the results after the 1 hour of rest. At time point 0, the sugar drink was consumed. After the 1 hour of shivering, the blood glucose levels were actually increased. This was in contrast to what we expected, as we expected the shivering to actually decrease the blood glucose levels. However, a similar response is also seen after exercise in healthy adults. It's important to note that the blood glucose measurement was performed immediately after the shivering. After exercise, blood glucose control is improved the next day, So we expected a similar effect with shivering. We expected the blood glucose control to be improved the next day. This leads to my next experiment. For this experiment, myself and Sten, we exposed 15 adults to cold. They shivered for 1 hour on 10 consecutive mornings. Before and the morning after the last cold exposure, participants performed the glucose tolerance test. Important to note is the differences before my previous experiment. This time it is repeated 10 times, the shivering, and then the glucose tolerance test was performed the day after, instead of immediately after the shivering. Here are the results of the blood glucose before the shivering, and here are the results after the 10 shivering sessions of the blood glucose control. We can see that the 10 sessions of shivering reduced the blood glucose concentration by a significant amount. This is an extremely important finding, as it suggests that shivering could be used as a therapy for type 2 diabetes. That was not all. Shivering also had other large benefits for health. Shivering reduced the amount of fat in the blood, which could be beneficial for preventing atherosclerosis, or fat buildup in the blood vessels. In addition, shivering reduced the blood pressure in all participants. So, alongside being beneficial for people with type 2 diabetes, the shivering could also be beneficial for people with high blood pressure or hypertension. Therefore, this experiment shows that the repeated shivering could be a novel strategy to improve health. For the second part of my research, I investigated the long-term adaptations to cold. This research will give us important insights if cold exposure was ever used as a long-term therapy. For this, myself and Wouter went to Tuva in Siberia in winter for 1 month, when the outdoor temperature was regularly minus 30 degrees Celsius. We performed research with a unique population. The population were Tuvan pastoralists living in yurts. Their livelihood was their animals. Some of them had over 100 animals which they needed to feed daily. For this, they had to shepherd them to pastures in the freezing temperatures for many hours per day. For 2 weeks, we attached sensors to the participants. In particular, we attached a thermometer to the participants. This showed us the temperatures that the participants were exposed to. Here is an example of the temperatures the participants were exposed to during the 24 hours. The black line shows the temperature the participants were exposed to. The red line indicates the maximum indoor temperature, and the blue line the outdoor temperature. As you can see, during a 24-hour period, the temperature the participants were exposed to was highly variable. On average, the participants were exposed to temperatures below 0 degrees Celsius for 5 hours per day. We also measured the energy used, or the calories used, by the participants during this 2-week period. For this, the participants drank labelled water, or special water, and then we collected urine samples before and after the 2 weeks. We measured how much of this labelled water was in the urine, and with that we can determine how many calories the participants used during the 2-week period. We found that the energy used per day by the Tuvans was significantly larger than compared to other populations, Europeans, for example. This is important as it suggests that the thermoregulation could play an important role in daily energy expenditure, which could have implications for obesity. As the Tuvans were exposed to the cold on a regular basis, many times we deemed these participants to be suitable to study the long-term adaptations to cold. So we compared the cold-living Tuvan population to 13 Dutch adults who were living in the Netherlands. We put them in a cold room at 10 degrees Celsius for a maximum duration of 1 hour. we measured how much energy the participants used, the shivering response, and also the blood flow during the cold exposure, and we compared the two populations. We found that the energy use and shivering response was similar in the two populations. This was in contrast to what we expected. However, it is promising to see that the shivering response and energy expenditure during the cold exposure are similar to the Europeans, because the shivering response and energy use appear essential for the previous benefits that I found in my previous experiment. A notable difference between the two populations was that the Tuvans had a higher blood flow to the finger during cold exposure. This is important because it will optimize hand performance in these extreme environments and also reduce the risk of injuries such as frostbite. This knowledge is very beneficial for populations exposed to extreme environments such as the military. To summarize, my first experiment showed that 1 hour of shivering did not improve glucose tolerance. However, repeated shivering did improve glucose tolerance or blood glucose control. In addition, it reduced the amount of fat in the blood and blood pressure. Thus, shivering could be highly beneficial for people with type 2 diabetes or cardiovascular disease. The second part of my research showed that the daily energy use of the Tuvans is high compared to other populations. This suggests an important role of thermoregulation in daily energy costs, and again could be useful for researchers investigating energy balance disorders such as obesity. The Tuvans and Europeans had a similar energy use and shivering response during the cold exposure. This is beneficial if cold exposure is used as a long-term therapy, as the energy use during cold exposure and shivering appear essential. Also, the finger blood flow during the cold exposure was high in the Tuvans compared to the Europeans. The main finding of my research is that the shivering appears very beneficial for health. Thank you all for your attention, and I give the word back to the Pro-Rector.
Thank you very much for a very clear presentation. It's now time to start the opposition, and the opposition will be opened by the chair of the assessment committee, Professor Mensing. Professor Mensing is Professor of Molecular Nutrition at Maastricht University.
I'm here. Thank you. Dear candidate, first of all, I would like to congratulate you with this thesis, and of course also your promoters and co-promoter. I think it presents very interesting outcomes. Studies have been well performed, and I'm pretty sure they were quite challenging, as we could see on your slides, -30, a little bit the opposite what it is now outside. And of course, I'm pretty glad I found a new argument in wintertime at home to turn down the heater at home. Always an argument between me and my wife. I think 18 degrees, 19 should be sufficient. Except for the scientific value, also the pictures in your thesis are very nice, but that is maybe due to the challenging environment you have been. When I read your thesis, I noticed that you made some comparisons between cold exposure and physical activity, and both are characterized by an increased energy expenditure. And my first question is it really cold exposure? Is there something special about cold exposure, or is it simply due to the fact that energy expenditure increases in your studies?
Dear highly esteemed opponents, thank you for your kind words and your question. I think it's something different. It's not just due to the energy expenditure. I think if you induce the same energy expenditure with physical activity, you wouldn't get as strong results what we have seen with the shivering. I think there are other processes contributing to the benefits than just the increase in energy expenditure. In particular, when you are cold exposed and shivering, blood flow to the muscles is altered. blood flow is reduced, and that causes, I think that will place different demands compared to physical activity, when the blood flow to the muscle is likely to be greater, more perfused, more oxygen availability, more glucose, more free fatty acids going to the muscle. So I think the shivering, the muscles will rely on more endogenous substrates, so muscle glycogen, intramuscular triglycerides.
Okay, so it is not simply the increase in energy expenditure?
No.
Okay, and then just out of curiosity, in chapter 2 and also in chapter 3, you did see a decrease in plasma volume. Can you explain it?
Yes, so yes, we measured plasma volume indirectly with the hemoglobin and hematocrit. This reduction in plasma volume is also seen during exercise. It's due to the increased blood pressure, forcing the water in the blood out of the capillaries. And also with the muscles, I think the osmolality will change in the muscles, and then water will be diffused into the muscle also. Also, perhaps there is increased urine production due to the increase in blood pressure.
But so you say, okay, it is due to an increase in blood pressure that causes decrease in plasma volume.
That, yes, and the muscle contraction.
And the muscle. But the decrease in plasma volume, is it not really a little bit of a problem? I did see that you adjusted your analysis for plasma volume changes, but still, don't you think that may make your interpretation of some of the results, especially when you talk in terms of concentrations, more difficult?
Yes, definitely. Care needs to be taken when you are correcting for changes in plasma volume. I think there's an argument for correcting or not for correcting for plasma volume changes. You could argue that correction is not needed because the absolute concentration is what is important. That is what the cells in the body are exposed to, the absolute amount. But correcting for it could be important for trying to understand why certain substrates increase or decrease. So I think that's the main reason for performing the correction. For example, we found, triglycerides, they are increased by a small amount. You may hypothesize that increased release by the liver, but because of the plasma volume reducing, perhaps it's just due to the reduced water.
But would you prefer to correct or not to correct?
I reported the main results uncorrected.
Without,
So uncorrected I would keep.
Okay.
I would present those.
You already mentioned briefly triglycerides, and maybe you know I'm a little bit interested in triglycerides, and maybe you are prepared now, because I hope so, because the decrease you observed is really substantial. But your study was 10 days. Do you think that is sufficient to make such strong statements about the effect on triglycerides? A 10-day study? Yeah. Are you sure that they are in a new steady state? Or do you think maybe I should be a little bit more cautious? And
I'm confident in the results.
Yeah, but also that it is stable after 10 days.
So
triglycerides. For example, I know if you do a dietary intervention, they always tell me, oh, it should be at least 14 days, otherwise you have not reached a new steady state. So your study was 10 days.
Yes, so then they may rebound.
Maybe,
Are you thinking? I think if the shivering was repeated continuously, then I think assuming the dietary the diet is maintained compared to before the intervention, assuming physical activity is also maintained, then if you keep repeating the shivering, I am sure these blood triglycerides would remain lowered.
Okay, so that's a huge effect, and then the question is, what can explain that huge effect? I did see in the general discussion, you said, okay, it may be due to the clearance. But when I looked in chapter 3, if it is the clearance, I would also expect an increased fat oxidation. But in chapter 3, you say, okay, substrate oxidation remained similar.
Yep.
Is that not a kind of contradiction?
So I think that, not necessarily. I think with the, so I think the triglycerides are just being stored in the muscle, for example, rather than being oxidized by the muscle.
Yeah, but you cannot continue storing triglycerides in the muscle otherwise.
Yep. So yes.
So you should be reaching your steady state in the end.
Well, there's the muscle, but other organs, perhaps the white adipose tissue, is also adapted. The liver.
Yeah, but still, in the end, you should have reached your steady state, and then you expect that at least fat oxidation, unless it goes to the adipocyte, maybe, but that it should increase. But you didn't see it in chapter 3.
Not in the morning when we measured the...
But in the morning you measured VLDL triglycerides, that are the fasted triglycerides. I would argue that it is due to synthesis.
Yes, also, yep. Reduced release by the liver.
Yeah.
Yep.
But can you think of a kind of mechanism that explains the reduced synthesis?
Yes, so we also found reduced free fatty acids. At the same time point as the reduced triglycerides. So perhaps the reduced free fatty acids, less free fatty acids going to the liver, less conversion into triglycerides and less release by the liver.
Yeah, I think that the free fatty acids are the driving force for VLDL production and not necessarily triglyceride production in the liver, but VLDL output. So maybe you can think about the possibility that also the synthesis of VLDL and the output of VLDL is decreased during cold exposure. Anyway, thanks for the questions, really impressive studies, and I would like to give the word back to the pro-rector. Thank you.
Thank you very much, Professor Mensink. The opposition will be continued by Professor Hopman. She's Professor of Integrative Physiology at Radboud UMC in Nijmegen, and she was also a member of the assessment committee. And Professor Hopman is present online. Welcome.
Thank you, Pro-Rector. Dear candidate, I would like to congratulate you with your excellent thesis, and in my congratulations, I would like to involve your whole team. It's a highly interesting thesis with high clinical relevance and also very surprising, especially part 2. For me, it was a very interesting and surprising part. And of course, with all the photos, the cover, I assume it's all, and I heard, in South Siberia. I don't know if South means, a lot warmer, but it's still Siberia, I can tell from the numbers you have in your thesis. So I've read it with great interest, and I have many questions, but of course I will limit myself a little bit. And so I would like to start with a couple of questions on chapter 3. And well, in chapter 3, you study the somehow long-term effect of the shivering, so 10 days, 1 hour a day shivering. And one of the effects that you see is, to me, a tremendous drop in blood pressure. Systolic blood pressure drops by 10 millimeters mercury and diastolic by 7, which is in clinical terms huge. As you state also, I think in your general discussion, it's associated with a drop of 20% in cardiovascular events. So that's enormous. But I could not really find in your thesis what your thoughts are about the mechanism of this drop. You mentioned that it's most likely not the plasma volume decrease, because that's a transient phenomenon. But what are your thoughts about what the mechanism might be behind this blood pressure drop?
Dear highly esteemed opponent, thank you for your kind words and your question. Yes, it was it was an interesting finding, especially the large decrease. It was unexpected. The main focus was the glucose metabolism. So when we saw these results, very interesting, and we didn't really set up the study to investigate the mechanisms in depth. But thinking about what affects blood pressure, cardiac output, and peripheral vascular resistance, I would think perhaps it would be logical to think along those lines. So for the peripheral vascular resistance, the radius of the blood vessel, perhaps that is more dilated. So with the shivering, it will induce it causes strong vasoconstriction. And then perhaps the next day there is a compensation where some blood vessels are more vasodilated. Perhaps this is due to the muscle. Maybe the blood vessels in the muscle are more dilated, allows substrates to be replenished in the muscle, for example. So yes, perhaps more vasodilation. In addition, there tended to be a decrease in heart rate. It wasn't significant, but perhaps with more participants, maybe that would have been significant. So perhaps reduced cardiac output. The basal energy expenditure did not change. In the morning, we measured the blood pressure. Yeah, so I think my main line of, I would think it's something to do with the vasodilation, more vasodilation.
Mm-hmm. Yeah, and could it be sort of a sympathetic effect in line with this somewhat, trend in heart rate and so a sort of sympathetic decrease? Is there something in literature related to cold and sympathetic decrease?
It would have been nice to have measured adrenaline and noradrenaline to investigate that. Perhaps, yes, perhaps. Yes, I think it's plausible. I would think, yes, plausible, I would say.
Do you expect it to be a long-lasting effect? I mean, did you see it in the pastoralis that they had lower blood pressure, or is it known that people in cold environments have lower blood pressure? I know the opposite, that people in warm environments have.
With the pastoralists, we didn't see. Yeah, that was difficult because these pastoralists, the men were highly active, many hours per day shepherding. So perhaps their blood pressure could be attributed to to increased physical activity. Yes, it's difficult to say with that, with the pastoralists, I'm afraid.
Okay. Then my second question on chapter 3 is a sort of less beneficial finding, I think, is that you see, well, there's a body mass loss, which is nice, 0.5 kilogram, but you also show that the fat-free mass loss is about 0.5 kilogram in 10 days. So that seems quite a lot. And well, in these already sort of metabolically compromised participants, what do you think of that? Is it a true finding? Is it why would you expect in cold, especially when you shiver an hour every day, why would you expect to lose fat-free mass? How can you explain that?
That's yes. Thank you for pointing that out. It's yes. So with the measurement, the body composition was with air displacement plethysmography, and then with that, as you mentioned, it measures fat mass and fat-free mass. So it measures the fat mass. We don't know whether the 0.5 kilograms on average of fat-free mass lost was actual muscle mass, or perhaps maybe water loss. So yes, I think that requires further investigation to determine if it is actual muscle mass lost. And if it is muscle mass lost, then perhaps the shivering intervention should only be used in maybe non-sarcopenic prone individuals. But if it is a true reduction in muscle mass, I know a colleague, Cas Fuchs, has done some research investigating the effect of muscle protein synthesis and cold water immersion. And then he shows that with athletes, following after the cold water immersion, muscle protein synthesis is inhibited or reduced, probably due to these changes in blood flow. So yes, it could be a muscle mass loss, but I am unsure exactly if it is or not.
Yeah. well, thank you very much. That's very insightful. Then I want to go more to a real lifestyle intervention. But before going to that, I was wondering, is it such a good idea to expose individuals to cold, either shivering an hour or maybe hours a day and a little bit milder cold? Because I started to doubt about it when I read chapter 4 in your in the pastoralis, and you show, at least I see in this table on page 90, that the women who are 51 years old, so I think that's not old, they have a BMI of 27.4 and a fat percentage of 38.9. So that seems to me rather high. So that started I started to wonder whether it's such a good idea to use cold to fight obesity.
I think cold for fighting or treating obesity is, I'm cautious with that. I don't think it can treat obesity. I think it has other metabolic benefits, such as improving glucose metabolism, lipid metabolism, high blood pressure. Just some anecdotal evidence from me is that when I'm cold, I am hungry. So perhaps cold exposure can increase appetite. I don't know, that requires further investigation. But the pastoralists, the men, they were highly active. The men and the women had a different lifestyle. The men were more active than the women. They were out for many hours per day. Then the women used to take care of their yurts more, maintain their home more. And the food, so probably they were had a positive energy balance compared to the men due to their diet. And if the women ate the same as the men, and they had the same food, and then they performed less activity, then yes, that could explain the obesity.
Okay. So maybe my last question. Is that allowed?
Yes.
Yeah.
Short question.
Yeah, short question, which is related basically to what you observed in this man. So while reading the whole thesis, I was wondering, wouldn't it be the holy grail to combine cold and exercise? And apparently the men in Siberia, they already do that. So what do you think of that? And is it do the beneficial effects run via different pathways?
Yeah, that was very interesting. That's what I said for future research. Need to investigate whether it's synergistic, additive, maybe even antagonistic effects. I think with the shivering, well, with the exercise, for example, you focus on, it depends on the exercise. Resistance exercise, you focus on certain muscle groups. I think with the shivering, you can activate muscles that are quite difficult to activate, maybe in everyday life. Such as deep core muscles, the recruitment of the muscles as well, with it being involuntary, perhaps that has a different effect than voluntary recruitment. Yeah, I cannot say for certain, but based on what I said to the previous opponent about the different pathways, I think there is something special about the shivering. The exercise, especially with the blood flow, vasoconstriction of the blood vessels, differences in substrate utilization. There is something different. It's future research to figure out whether these are complementary or not. But yeah, I think maybe the shivering and maybe resistance training would be useful maybe if it is a true muscle mass loss.
Okay, well, thank you so much for your answers.
Thank you, Professor Hopman. The opposition will be continued by Dr. Blondin. He was also a member of the assessment committee. He's assistant professor at the Université de Sherbrooke in Quebec, Canada.
Thank you.
Okay, go ahead.
Candidate, I'd like to commend you first for the tremendous amount of work that was completed throughout this thesis. I think This is very important and very difficult and demanding studies to perform. I think the candidates should be commended for all their hard work, particularly during a pandemic. And I think it has nice complementary work between work that's done in the lab and field work. I think it's a really important and nice contribution to the field. So congratulations for that. I have a few questions. First, starting with chapter 1, or sorry, the first study. We noticed here that the total energy expenditure during the 1 hour shivering was reported to be 619 kilojoules, but there was quite a large range between 451 kilojoules to 735. This is quite a significant range. What, if any, do you expect the impact to be on the individual responses following the cold?
Dear highly esteemed opponents, thank you for your kind words and question. Yeah, so for the first experiment, we individualized the cold exposure based on shivering. We individualized it based on primarily observation, and then we also used surface electromyography and real-time energy expenditure changes to sort of confirm that. But we didn't use any predetermined criteria for that. The shivering response is diverse in individuals, so that probably explains the range in cold-induced thermogenesis during a cold exposure. How it affects the results. Yeah, see if, yeah, that, yeah.
In other words, would you expect someone with a higher energy expenditure to have a different response than someone that has a lower energy expenditure? Would they clear glucose better, or would it be worse? Because here you're showing glucose especially is worse immediately after the cold. So what would you expect to see?
I would have expected with a higher shivering more cold-induced thermogenesis, more shivering, more muscle contraction. I would have expected improved glucose tolerance, so reduced blood glucose levels during the OGTT. That, compared to individuals who shivered less, had a lower cold-induced thermogenesis. But the response that we see is that the 1 hour of shivering actually increases the glucose levels during the OGTT. And then again, this seems to be a normal physiological response, at least in healthy adults, immediately following activity, so exercise, or also for the first time we show that in shivering. Then the mechanisms, increased glucose release by the liver, perhaps the muscle blood flow had an effect, less blood flow to the muscle, less muscle glucose uptake. So yeah, the cold exposure and the shivering, some things will reduce the glucose concentration, some things will increase it, and in this case, the mechanisms that increase glucose concentration prevail.
Now, one of the other things that when we look at insulin sensitivity, there's a number of different ways of looking at it. You can either look at it using an OGTT, you can use a meal test, or you can use the two-step clamps, which is obviously something that's used quite a bit in Maastricht. How do you expect these results to be similar depending on whether depending on the test you use, or do you think it might be test dependent? And I say this because we know that with OGTTs, we often see reduction in muscle perfusion, which is kind of paradoxical. You don't expect to see that, but it's a kind of a unique feature of OGTTs versus like a meal test where you don't.
Yes, it's test dependent, I would say. I believe if we performed a two-step clamp immediately after the shivering, I would expect muscle glucose uptake that to be shown that the glucose infusion rate would be increased, because the insulin would promote the supraphysiological insulin concentrations would promote maximal vasodilation. So then I would expect that any reduced blood flow to the muscle, that wouldn't be important. So I would expect it's test dependent. I think if you did a two-step clamp, you would see an improvement in insulin sensitivity or muscle glucose uptake.
Okay, so it is very much test dependent. So, and then I guess this is the tricky part about trying to determine is an improvement or looking at changes in insulin sensitivities. It seems to be very test dependent. And then an overarching theme, so a separate question here, an overarching theme that we see in the thesis, and I think it's an important theme that in the lab is considering the importance of standardizing the cold. So in some cases, standardization is very useful, and in other cases, it can sometimes mask effects. So would you expect to see different results if participants were exposed to the same, but like a moderate cold? So what if you were, rather than sort of standardizing for the shivering duration, if you standardize for shivering intensity, or if you standardize for cold-induced thermogenesis, or if you standardize for the rate of heat loss, would you expect to see similar effects, or do you think that sometimes it's a design feature?
Yes, I think ideally you would standardize for energy expenditure and shivering intensity, and that would be the ideal. I think that would be more ideal than standardizing for shivering duration, but I think it's for this experiment or future experiments, it would be very difficult to standardize for the energy expenditure and shivering intensity, unless actually you could do standardization test beforehand to see someone's shivering response. It's just extremely variable. Is the shivering response between individuals extremely variable? It depends on an individual's morphology, but then also as well, we noticed during the 10 days of the shivering, participants' shivering response also changed during the 10 days. Sometimes different muscles were recruited. Sometimes people were shivering more intense at the end of the 10 days. Sometimes people were shivering less intense at the end of the 10 days. So I think ideally, yes, I would standardize for cold-induced thermogenesis and shivering intensity, but it's extremely difficult. So I think we did the best thing possible, and then we made, in chapter 3, we made it reproducible, so other scientists could replicate the experiment and based it on when a participant's energy expenditure increased by 50%, then it lasted for 1 or more hour. So I think other scientists can reproduce that to try and replicate these findings that we have observed.
Yeah, and I think that's great. I think if you're able to reproduce it between investigators, I think that's really important. I think, like you mentioned, it can be very variable between individuals and even within individuals from one day to the next. I think it's important. And the other thing is, I think to keep in mind is just that attempting to standardize certain things in a certain way can also often mask differences that might be relevant. So I mentioned this because, for example, in one of the when you make the comparison between the individuals from the Netherlands versus the Tuva population, you report similar cold-induced thermogenesis and shivering between the two groups, but I wonder how much of that is because of the design itself. So because there was a deliberate attempt to match for those outcomes, then you're going to see similar results. But in reality, if you just expose these people to the same cold, so if you put everyone to 18 degrees or 15 degrees outside, do you expect to see that the shivering and the heat production will be the same between individuals, between the two groups? Or is it a design feature?
I think so. With that experiment you are referring to, with the Tuvans, yes, we individualized it based on the Tuvans' shivering response, and then when they started to shiver, we added an extra 10 minutes on to see their response. Then the participants were individually matched based on their morphology, age, and sex. Well, their height, body mass, age, and sex. And then the time duration for the individually matched individual was the same. So I think that comparison is fair to do, but still, if we use a different test, I would expect different. Yeah, I could foresee different results if the cold exposure was, so we used 10 degrees Celsius air. If we used a more strenuous, a more severe temperature, a lower temperature, something that is more like the Tuvans would be exposed to in their daily life, below freezing, then I would expect the Tuvans to be more adapted and perceive, and their body would perceive the extreme severe temperatures to be less threatening, so maybe less cold-induced thermogenesis, less shivering response. But then again, the study needs to be done because we also found this increased blood flow to the hands. Now, in a cold environment, that would actually increase heat loss. So that would increase heat loss compared to the Europeans who have greater vasoconstriction in the hands. So the study needs to be done, but there could still be differences, yes.
Maybe one quick question, if that's allowed. The shivering here we see across the sorry, is this okay? Yeah. So the shivering also is there. We see different definitions throughout the thesis. Some is based on visual detection, some on EMG, and some on changes in energy expenditure. Can you comment just briefly on what you think when we talk about shivering onset or shivering intensity, what is the most appropriate measure?
Yeah. I would say yes. EMG, shivering onset, shivering intensity. I would say EMG is the best method. I refer in my book. I mentioned shivering thermogenesis, so heat production with shivering heat production, and then yes, I think in chapter 3 we refer a lot to shivering thermogenesis, and then with that, I think a combination of EMG and the indirect calorimetry to measure energy expenditure is appropriate, but definitely for shivering, EMG is the best.
Thank you, Dr. Blondel. I'd like to continue with the next opponent, Dr. Meijer. He's associate professor at Maastricht University.
Thank you, Pro-Rector. Dear candidate, I also like to convey my compliments for your nice thesis and also your very clear presentation. It really helps me as being a novice in this field. Also, my compliments to your supervisory team, which you have spent, or have some great adventures with in Russia. I think an experiment that will be hard to replicate for the next decade or so due to political reasons. But I'm a bit jealous. I mean, doing a PhD and then actually doing a field study. I've spent most of my life in the laboratory. I guess this gives you a lot of adventures and bedtime stories for your future offspring. So that's already something you can take with you. I also impressed by your ambition. I mean, you're taking over from the pro-rector in terms of managing the questions. So that's well done. I'd like to follow up maybe on the question posed by your first opponent. I'm a movement scientist by training, so my bias is that physical activity is good for everything. You're basically proposing that there are alternative strategies, and I'm not fully convinced by your answer. Because basically your first experiment in chapter 2 does not allow you to make that inference, right? You're comparing a resting situation with a shivering situation. How could you improve upon that protocol to maybe firmly attest that shivering really triggers different mechanisms than physical activity? And you, in response to the previous opponent, you mentioned that standardization will be very difficult. I'm inclined to say it's, in this particular case, not so difficult. Yep. So maybe you can enlighten us with your answer.
Dear esteemed opponent, thank you very much for your kind words and your question. To really tease out the differences between cold exposure and exercise or physical activity on the effect of these metabolic parameters, the first experiment has a shivering session, a resting condition. We could also add an exercise test in there. I think the best, the way we could do this, we could use, I think a cycling exercise would be reasonable. And then we could also, and then we would measure and then I would attempt to match the shivering and the exercise on energy expenditure, or the change in energy expenditure. And then how we would do that, we would do the shivering test first, measure the energy expenditure all the way through, see how much increase in energy expenditure, or how their VO2 or energy expenditure if we have the equipment, see how that changes, and then replicate that on the cycle ergometer. Then we could add weights, remove load, increase load, decrease it.
Yeah, I think that I guess the nice feature of that is that managing energy expenditure during an exercise is easy to control, and the shivering, of course, is a very uncontrolled situation. So having the shivering before the exercise would be beneficial there. I have some other questions, so we'll not continue on this one. I would like to ask one of your opponents, or paronyms, sorry, to read out proposition number 6.
Separating shivering from non-shivering thermogenesis is difficult and problematic still to this day.
Okay, now I noticed that this paper is in 1963. This is older than I am. I hope there has been some progress in this field, but why is it problematic?
Yes, as I mentioned before, it depends how you define shivering. We can see it's very objective to measure shivering or define it or see it happening with EMG, surface electromyography. But specifically, I'm referring to the shivering thermogenesis and non-shivering thermogenesis. So the energy expenditure, so the heat production, energy expenditure due to the shivering and due to the non-shivering thermogenesis. I think quantifying it absolutely is rather difficult, because I suppose it depends on your definition of shivering thermogenesis and non-shivering.
So is the problem to objectively identify shivering? Or is the problem to objectively identify the energy expenditure due to these two different mechanisms?
Yes, exactly. Okay. Quantifying it due to these two mechanisms, because I think sometimes they occur in parallel. When it looks like an individual is not shivering, they could be shivering due to the recruitment of deep muscles. We cannot place surface EMG all over the body, and we cannot measure all of the muscles, especially the deeper muscles. So I think I would be very careful to try and quantify these absolute contributions of shivering and non-shivering. Yeah. And I think I would,
Okay, I'm satisfied with that answer. With respect to the shivering, are you familiar with the paper by Arnold in 2020 in European Journal of Applied Physiology?
Josh Arnold, I think.
Yeah, they tried different techniques, and they concluded that in the end you probably should have a combination of the mechanogram, the electromyography, the energy expenditure. You mentioned that you also had these three elements to make your assessments, and you mentioned that EMG was the best, but why is EMG better, or why do you feel that EMG is better? Because if I look at that paper and I look at the distributions in the data, it's all over the world.
Yes. I suppose it depends on what your aim is. With the EMG, we know that participants were shivering. I think that was the aim of the experiment, to really show that participants were shivering during the cold exposures. With energy expenditure changes, I suppose we can assume that it depends. You may assume that participants are shivering with the energy with the changes in energy expenditure, but
even with the EMG, they show that because you can have a sort of algorithm to identify the threshold in the EMG. You can also use visual inspection of the EMG, and so with the same signal, you might get different results depending on the criteria you use to define the threshold. So. Now I noticed that the pro-rector will shut me up in a few minutes, but did you use visual inspection, or did you have an algorithm to identify the threshold?
It depends on the, so for the Tuven experiment, we really looked at the onset, and for there we used an on or off with the EMG signal when it was 2 standard deviations above the resting, the baseline, the resting, when there is no muscle contraction, we counted that as on. And I think figure 1 in chapter 5 shows with the small arrow that criteria and how it looks with the EMG data.
Okay. I'm satisfied with your answers, and there are many more questions, but I should leave some time for my colleague. Thanks.
Thank you, Dr. Meijer. The opposition will be continued by Dr. Jongeres. He's associate professor at Maastricht University.
Dear candidate, of course, first of all, I also would like to congratulate you on this very, very nice piece of work. So in your thesis, you focus both on an acute and longer-term study, and you also compared some very, very nice results of field research in Siberia. So my compliments for that, and of course, I also would like to extend my congratulations to your supervisors. Now, due to time constraints, I would like to immediately challenge you with a question, and actually it's a follow-up question from the discussion we had before with Professor Hopman about the blood pressure results. Because in your thesis you also claimed that cold acclimation can be an alternative lifestyle strategy for people with hypertension. But did you consider the data presented in chapter 2 when phrasing these results? And especially I'm thinking about the blood pressure effects during cold exposure.
Dear esteemed opponent, thank you for your kind words and your question. So you are referring to the increase in the blood pressure during the shivering, and then how that links up with the improvement in the blood pressure after.
Yeah, or more that you state it's like maybe a therapy for hypertension, but so your study group already has grade 1 hypertension.
Yes.
So if you expose them now to shivering, what's your opinion about the safety?
Yes, that's a fair point. I suppose it depends on the extent of hypertension and the severity in the participants. Because the shivering does increase the blood pressure, so you wouldn't want maybe to do it in people with late stage, quite a high stage hypertension. So maybe it's suitable in these individuals. So I should have probably been more specific in which category of hypertension. So there is that. Then with the exercise, though, that also increases blood pressure. Also increases it to quite a high amount, especially high intensity exercise. Mm-hmm. And I think that seems to improve blood pressure and seems to be rather safe. So I think it's worthwhile progressing, but carefully, cautiously with this.
Okay, I agree. So, but also if you take into consideration that the increases in blood pressure, I think you already mentioned so, is probably due to an activation of the sympathetic nerve system, and that's due to shivering. So what would it be, for example, an option to state, okay, we should focus on cold acclimation without the shivering, because then you don't have these safety aspects?
Yes, that's a good question. So these the blood pressure results that we have after this shivering intervention, it's not been shown before. This is the first study that has really targeted shivering, made people shiver for at least 1 hour time, 10 consecutive days. So, the reduction hasn't been shown before. In the previous experiments, it's shown the cold acclimation with negligible shivering, some shivering, improved insulin sensitivity, but there was no change in blood pressure. So to me, that suggests that this intense shivering seems important for the improvement in blood pressure.
Mm-hmm. Okay, and if we move on a little bit to the mechanisms, because you already talked about it, like, for example, tissue perfusion is very important. And also you come up with I think I really liked that one. A measurement of vascular function. You also performed in Siberia to measure perfusion of the finger. So can you maybe explain a little bit what you did on that one, how to measure perfusion of the finger?
Yes, so in Tuva in Siberia, we measured the forearm fingertip gradient. We attached a temperature sensor to the forearm, a temperature sensor to the finger, and then that gives us an indication of blood flow. In particular, we performed a measurement called the cold-induced vasodilation test, and then with that one, we just attached one temperature sensor to the finger, and then there is a container full of ice water. The temperature is 0 degrees. The participants put their finger into the ice water for 30 minutes. We constantly monitored the temperature of the water, 0 degrees Celsius, and then the temperature at the skin of the finger would decrease towards approaching the temperature of the ice water, 0. At certain times, blood flow is increased to the finger, and we can detect that with an increase in temperature.
Because my question is a little bit, what are you really measuring? Is it tissue perfusion or maybe vascular function? Because maybe you call the cold pressure test. Did you ever heard about it?
Yes, with the hand, I
with
believe.
the hand in ice cold water. So that's a measure of endothelial function, and it's actually based on an activation of the sympathetic nerve system due to pain. Would that also be something to take into account? So when observing these data, because I think you showed some nice results on tissue perfusion in the finger, would that also be something that is all due to like an improved vascular function as a mechanism and not perfusion per se?
Yes, I think they are related, the vascular function and perfusion. So yeah.
Well, you can give the answer later on. Adam Jake Sellers, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Adam J. Sellers. The degree committee here present has assessed the quality of your thesis and of your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Mark van Marken Lichtenbelt is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
All right,
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Adam J. Sellers, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate, signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Here you are.
It's important. Okay. It's yours.
Where you can
You
look
can put
after
it
that.
there. Or you can Dear Adam, Dr. Sellers, a well-deserved congratulations. Great job. It's an honor for me to give the speech to you, and I do this on behalf of your supervisors, Professor Patrick Schouwer, who unfortunately cannot be here at the moment, and your co-promoter, Hanna Palabinski. It was good to have you here from abroad, from England. During your studies, you showed a strong interest in sports and exercise. Viewing both your bachelor and master studies in Loughborough and Leeds, for which you were awarded with several high grades. You did your master research on a project on the effect of chewing gum on soccer players. Maybe that is why you got interested in do a project here with us at nutrition and movement sciences. But no, of course not. That's not it. It really was your intrinsic interest in the physiology of humans. It could have been nutrition. It could have been sports. But it became thermoregulation and the effect of cold. During the interviews, you made a very good impression to us. You made a sound proposal and came over to Maastricht to give a presentation. It appeared that you really delved into the matter in a structured way. We've also made inquiries to an old supervisor, and he made very clear, he will be really good, I will take him. So that's what we did, and we didn't regret it at all. And indeed, you appear to be a very serious worker, always very well prepared to your meetings during the studies, taking great care about the actual measurements, always very precise and taking care of the details. Later, during your second large study, you also appear to be good in collaboration, and that was not at the start really obvious. The first study you did all by yourself, and that worked out well. So at the start of the second study, you had to get used to working together, but it went very well, and the collaboration between you and Stan van Beek, your paranymph, was fruitful and stimulating. So this was important because the studies were far from easy. It asked a lot of both of you as well as of the volunteers. As you have seen, All of you on the movie, you had to shiver for an hour and that 10 days on a row. So it was quite something. And some liked it, but some liked it, so to say, hotter. So, but you managed to measure all the 15 subjects, men and women, and in general, it went smooth. You did have a hard time during Corona, but despite the problems around this, in this period, as soon as we were sitting together and we were discussing and interpreting the data, I saw a change in your eyes, a change in your attitude, lighting up and dive into the matter as if no Corona or whatever existed. The scientific data grasp your attention, and I think that's typically Adam. A serious investigator and a critical one, and I want to give another example. One time we got a demonstration of a cooling instrument from a company who thought to get big money for cooling people in a hot environment, and it was made to really do cooling the hands, and it was an instrument with a lot of electronics and a nice display. So, and as long as there are electronics, people tend to be impressed. Just cooling the hand, and Adam tested the instrument. And Adam wrote a small but concise report of the test, and that clearly reflects his critical attitude. So I got this by mail, and one of the two sentences are as follows. I think any performance effect they have seen is just a placebo effect. For example, a red pill gives a better placebo effect than a white pill. I think the same is true here. A large piece of technology produces a greater placebo effect than something as simple as cold water in a bucket. Period, clear conclusion. So we didn't really continue studying with that instrument. In some things you can be a little bit more stubborn. I remember that your first presentation, which was to meant for a meeting, for a symposium of 8 minutes. And then you showed the first slides. You showed the slides to Hannah and to me, and it appeared that you had 30 slides, to mention a few. And throughout the years, that simply didn't change. Every time you gave a presentation, you started with 3 times too many slides. But at every time, in the end, your presentations were still very well, and we saw this with this defense, but even for this defense you had, I don't know how many slides, but
40.
And in the end you had a very nice presentation, and the committee also agreed that it was very clear. When I asked you to join in a quite exceptional field study in Siberia, you didn't think twice. You were delighted to take part. I had prepared a general study design, but together we worked out all the details, add other tests and the instrumentation, the transport, the pyramids. And because of a lot of time pressure, it was quite a job to get things set, and only a few hours before the departure we were really ready to go. A great joint effort which continued in Tuva itself. There, together with Dolana Kovalik, who facilitated the studies, and our PhD student, we were a great team and an exciting time. Hard work, sometimes exhausting, but often very satisfying. If you read the acknowledgements in the booklet, you will find some more details, but I will provide also some. But it really shows how much you liked this experience. But things changed when the Russian FSK got behind us and tried to kick us out of the country. This gave us sleepless nights, early morning travels to Kyzyl, the main town. I was worried about the data that still needed to be gathered, but Adam was more worried about his freedom. I joked, they cannot send us to Siberia, which at that time, you, Adam, you couldn't really appreciate that. But even then, there were positive highlights. I remember that we had to go again very early in the morning to Kyzyl for the visa. We were all rather sad, unsure what would happen in town. We got some extra time and visited a local museum. Then we were so lucky to find out that the large national Tuvan orchestra was practicing in the cellars with the local instruments like the 2-stringed harp, the drums, and the throat singers. We were so lucky to be able to join and were introduced to the band. We were tired and tried to keep our eyes open. Then comes the first song. Immediately we got overwhelmed by the power of the music and the intensity with which it was played. All the strings bow in a slow and compelling rhythm. The throat singers, everyone falls in. Shivers run down my spine. After all the misery and the uncertainty surrounding our stay, the worries about us and the research, the sleepiness and the sad mood suddenly gives way to relief. And later I heard you had exactly the same experience. From then onwards, things went almost magically smoothly again. Thanks to Delana and her family, we had a once-in-a-lifetime experience. I'm very happy that you continue the hard work of Tuva after the return to Maastricht. You did a lot of additional measurements and managed to write the papers we planned for this study. They are now all published or accepted for publication. Great job. I would like to thank the reading committee. Where are you? There's the screen. Especially those from outside Maastricht, Denis Blondin, all the way from Canada, and Professor Maria Hopman from Nijmegen. Thank you for attending via Zoom. Adam, 2 days ago, when I prepared this laudation, I wrote something like, I hope you will find a job. And now I can say, I had to rewrite it, I can say, you got the job. So congratulations.
Thank you.
Very nice that you got it and that you finally can have this in Amsterdam. And I hope you get an interesting and inspiring work during that job. I would like to end with the congratulating your mother and Jolien. And now it's party time.
Just a few more words before it's party time. Dear Dr. Sellers, also on behalf of Maastricht University, I congratulate you with the degree you have acquired. I also congratulate your family, friends, colleagues here present and maybe online. It's the second time we meet this week. Yesterday you were the paranymph at the PhD defense of your paranymph, Louis Wij. And you told me afterwards that you probably were more nervous for his PhD defense than Louis Wij himself. I hope today it was the other way around. Yes. That you were the nervous one and that you felt okay and on top of everything. I can tell you that the committee was really happy with the way you defended your thesis. So no worries about that. And I hope you also feel satisfied with the way you defended the thesis and relaxed about everything now. I'd like to thank the members of the degree committee for their effort and time to make this PhD defense ceremony a success. Thank you for the people online for your contribution. I'm sorry you can't be part of the better times afterwards, so to say, but thank you anyway. And with this, I'd like to close this academic ceremony.